I will present you some work mostly done at when I was CNRS in Paris, which I now on leave, and I'm in Milano at Politecnico. So, to cut it short, this talk will be about shape. And shape, it will be what mathematics can explain about the generation of shape. So, it is a problem of has attracted much of interest over the history, especially for the common man. When the common man sees all these kind of nice patterns organized, it's kind of sees a divine intervention in that. So you imagine all these golden ratio patterns that we find in nature. They inspired even a book in the 16th century. It was called The Divine Proportion. So we really thought it was something about God putting a shape into stuff. Then that's where the atheist mathematician comes into the picture, because the atheist mathematician comes and says, well, that's just self-similarity. So that's maybe that some governing equations there admit a self-similar solution, like it's, for example, for the phyllotaxis in the sunflower. It is just an optimization of coming the next seeds in order to optimize the food that it gets. You see, even we have some kind of putting to an extent this kind of speculation even in painting, in architecture, and so on. But actually this kind of religious scientific debate went on over the centuries in history. Even we have to wait till the 17th century till someone told that actually shapes is something that evolves over time. And it's even strange if we think that even when after that the microscope had been invented, we find in scientific literature this kind of pictures, but they still couldn't get into looking at what's inside the sperm cell. But they imagine they found this nice little lady, was a nice reproduction of an adult, or this little homunculi, as they called, that were ready just to expand as it was an affine transformation in there. So actually there was this dichotomy going on over the century. From one side there were people saying, okay, religious viewpoint, that's God putting us there, it's preformationism. And the other part was epigenetics, and said, actually we evolve. That's the most famous statement is from William Harvey, which just, it's not something more original, because it was, if you look at it, it was already in Aristotle's generation of animals, which actually ex ovo omnia. We all come from an egg, but we all differentiate for that. So actually shapes is a property which is adaptive. It's something that depends on the environment. So at one point this all went over the years. We have even a mechanicistic epigenesis, which we arrived with Claude Bernard, who actually said that the matter possesses a state of mutual equilibrium with the environment. And this mutual equilibrium has to be robust enough, but has to allow adaptation to a change of the environment. So he actually called it, it was a little mysterious in his definition, but it was some internal matter that was balanced by the external matter. How this balance was achieved, it was not in Claude Bernard's words, but it was actually the birth of the modern sea of embryogenesis. After that, we see in the modern picture, we passed through some milestones, and the milestones are, for example, Darcy Thompson. We are in the right place to talk about him. It was his only masterpiece published by the Cambridge Press, which has actually demonstrated something that was known to Galileo already, which was the fact that the shapes of some animals actually have an allometric scaling, which represents some sort of a static diagram of forces which act on them. Then these static viewpoints can have evolved, and the evolution came with Alan Turing and with famous Turing Instadilia, the fact that chemistry was important, so diffusion is important. So actually static patterns can come from a dynamic equation involving reaction and diffusion in certain limits that we all know. Then actually there was a kind of development in the biological picture, and what Turing called morphogens, not just as he actually extended a concept made by Waddington before him. It was like morphogen is something that evocates a form, a shape. Actually, it was finally found scientifically many decades away, which is something, some kind of signal, biometrical factor, whatever, which possesses two properties. Can induce growth, and can give positional information. So whatever protein, growth signal has these properties, it can be called the morphogen. And the simple idea of the morphogen is the French model of Louis Volpert, which is a sink cell here, which is pink, a source cell which is there in green. It creates a gradient, and there are threshold values, and if the gradient is if the value of the morphogen is in this range, it develops the blue pattern, then it can, otherwise the white, otherwise the red. So extending it in much more complicated diffusion reaction models, we can have all the flags in the world. So that's a picture which actually got kind of mainstream for a lot of centuries, but which actually misses an ingredient, because this is all based about chemistry. So we go back and say, okay, now we are in the modern picture. Then genetics comes through, and then somehow we see again this dichotomy. Because genetics at one point said, okay, but we discovered there is DNA. DNA has all the information. So what happens is just, it's the information there. There is some kind of machines that put the algorithm, it may run it, and it gets you the final shape. So it's like a modern view of preformationism. Everything is already inside the genes. Opposed to that was the fact that actually there is some kind of epigenesis, which means that if you change the external factor, you can change the evolution and the shape of one organism. So actually what we stick to is to the fact that we call the morphological evolution as all the phenotypic change which in the development of the single organism which happens during the developmental of the species. So because we have only the genotype, which is a subset of all the genes that we possess. So that kind of genes can change expression in the single individual. And then we look back at what Turing said in his paper. And what Turing said was like, when we have to go and say, look at the shapes and stuff, we have to put Newton's law of motions, because that's important. We have to account for stresses, because everything that gives a motion can actually, its vocabulary is just the elasticity. So that's the way he expresses his feeling about the outer world. We have to take into account chemical reactions, diffusion of chemical substance. Once he makes these very good statements, Turing himself says these things, that interdependence of chemical and mechanical data adds enormously to the difficulty. So a solution could be easy when one of these two cases can be separated. And then he keeps on doing his chemical basis of morphogenesis, which is actually the title of his book. What I'm trying to go is just to make a step forward then and see how we have to postulate the chemo-mechanical basis of morphogenesis. First of all, as a mathematician, I know that we are all sensitive to one argument, non-uniqueness. And the source of non-uniqueness in morphogenesis is just the vocabulary. Because if a biologist comes to a mathematician and say, okay, cells differentiate, the mathematician thinks that they are able to calculate the derivative. And he thinks, oh my God, why the cells do and my students don't? But then it seems it's kind of a funny way, but it's actually a big barrier in between who does mathematics and who does experiments or biologists. So even in the future, when I think about stress, don't think of that huge amount of email that is waiting for you when you finish this. It's just mechanical stress. So just few useful definitions. I will stick to morphogenesis as all the ensemble of all the possible different origin processes, biological, physical, chemical, that have as a final result a change of shape. So I will refer to growth as everything which is concerned to a change of mass, remodeling it everything which is concerned as a change of microstructure. Differentiation is when a tissue actually cells changes and it changes type. And especially I will focus on pattern formation from a mechanistic viewpoint. So all in that cases where the mechanical arguments are important. There is always a huge debate about it, but it's sometimes mechanics is just a pre-patterning for biology. Biology uses mechanics. It's not mechanics that explains everything, but it's a tool that biology uses somehow to facilitate its task. So when we're dealing with living matter, we have to be concerned by few important things, which is the fact that compared to inert matter, we are in a range of lower typical scales. We have softer materials with complex biologies. There is still a huge debate if you go into literature who uses liquids, solids, viscous fluids, or viscous solids. I think the most paradoxical question is if solid tumors are liquids or not. I mean, it seems strange, but you even find a lot of papers where solid tumors are treated like fluids. So I don't know if it's kind of a vocabulary that we all have to start building together with biologists and medicals. I think there is a problem in the base. If we want to really create a generation which speaks the common languages, we have to start from making a school where doctors and mathematicians teach together, so they agree on a vocabulary first. So what's the modern picture of morphogenesis? I think this slide that I took from a review paper of Lequitt and Legoff, it's kind of explicative in this sense. So at the beginning, let's stick in the embryo, which is the easiest way of speaking about morphogenesis. There is just a bunch of cells. And in these bunch of cells, there is random cell divisions. Why random? Because it's not organized. There could be a mutation, there could be some kind of noise, whatever. It's just a stochastic process. At one point, some polarity goes in, and that polarity is just smells a little bit like those morphogen stuff that we talk about Voltaire, the French French bodies. There is actually a polarity that comes from chemistry, and this polarity orients cell divisions. At one point there is a differentiation, so there are different subcellular types, and they are in competition to each other. At that point there is growth, there is competition, but there is even rearrangement. What does it mean? It's called, for example, intercalary growth, which is the fact that cells actually change their relative positions, so the structure changes. At that point, so imagine that this tissue is growing. At that point, there is a morphogen that in one certain range domain actually creates growth. Growth goes and creates a compression from the outer tissues. What is known as this compression introduces an inhibition of growth. So actually, this inhibition of growth, mechanical, creates a variation in the chemistry because the concentration of the DDP or the morphogen is changing. And at this point, they actually have a kind of a feedback in between the two, and the final result is what we call homeostasis, which means that they find a steady state solution where everything is in balance, chemistry and mechanics. So Turing was right. I'm not going to go very much further on what it did, but there is actually the need of going through on adding mechanics to chemistry. So first step is just adding balance equations, and we add it from the mechanics. So you consider this as an embryo. This embryo has cells, as morphogens, and morphogens here are small weight molecules, so we can neglect them in their inertia. We can consider them as internal variables of the problems. So there is sources, volumetric sources of mass. There is non-convective mass flux. So actually the mass balance equation has these two adding terms. And these terms actually can depend on these morphogens. Okay? And these morphogens actually have a classical reaction diffusion equation. There is something you can diffuse, they can react. Where they are, and what would you like to say is just that wherever the morphogen has an uptake, it creates an increase of mass. And this increase of mass can happen volumetrically or in a surface way. So then we have to add the momentum balance. We have to take extra care about the non-convective mass flux because it introduces some extra terms, and then we want to add thermodynamics in that and say, okay, but we want to do something which is actually consistent. I don't want to create mass from nothing. I want to create energy. I actually want to have a model in which this exchange between chemical, mechanical, it's thermodynamical consistent, meaning it dissipates. So actually we have some constraint given by the Clausius-Duhem form of the inequality. There is a specialization of this kind of theory whenever we speak with soft solids, so we can actually create the fact that there is a reference configuration. What is that? Is that the reference configuration can change because it can grow this volume, can change its microstructure. So actually it depends explicitly on the material point. So depending explicitly on the material point, we have to understand how to set this dependence. And one way of doing it, it's just something borrowed from the theory of plasticity, just introducing the fact that actually every point here could evolve into a natural point, which is not necessarily a physical point. It's just that it can have a different natural state that can evolve over time. And then there is actually an elastic deformation that restores it the physical compatibility. So actually the elastic model, which is this part, is just typical elastic behavior. But there is a change of density, which is given to the fact that here there is a change of volume. So actually we have a tensorial dependence on the natural state. So we change the natural state, but we keep on behaving it the same on a mechanical viewpoint. So if you add that, we have the fact that we can introduce that this term here, which actually says which is the rate of growth, I don't want to go too much into details, is coupled to a stress, and this stress is just the Eshelby stress. Those of you who are familiar with singularities or whatever pitch color forces or whatever you call them, you know that the Eshelby stress is just the conjugate. If you project everything into the material manifold, which the Eshelby stress is the stress conjugate of this moving microstructure. So whenever there is a microstructure who moves in the Lagrange, it's energetically conjugated with this stress component. We can even push this theory to an upper level, which is like not only we make this linear map of tangent spaces, which was this decomposition, but we can make a two-jet composition law saying that not only it changes its volume, but it also changes the curvature around it. So actually you restore it and you have what you call the first order and second order connection on this stuff. And what you get is just the first order connection is just the torsion of this material connection, while the second order connection is the curvature, as we and both of them have this stress conjugate related to the Eshelby stress. But by doing so, and we are plugging in these results into the Clausius-Duhem inequality, we understand that actually if we want to construct a stress-dependent growth law, the stress dependence has to be the fact that the Eshelby strain conjugated with these tensors has to give a positive result. So this allows us to create kind of remodeling laws from the material which is thermodynamically consistent. I know it seems kind of abstract in that. I'll try to give you just an easy example, which is tumors. We all know that if we take a tumor spheroid, and there was some pioneering experiments in that group made by the group of Rakesh Jain in the 90s, now redone a few years ago in Paris in other ways, but actually the point is the same. If this cancer spheroid grows, and it grows in agar, for example, or under an osmotic pressure, which is compressive, actually what we observe experimentally is that first you have what you expect, the exponential growth, then you have some kind of saturating regime, a linear growth, and then actually if you apply too much stress, it's just growth cannot happen because it's no longer thermodynamically consistent. If you just do the same stuff here, you can see that there's an avascular tumor that grows, compression over the external tissue. you apply that into that evolution law, so you take care of that stress-driven growth law using the Eshelby stress, you actually are able to create these saturating growth laws. What does that mean? It means that the longer you spend in growth creates some kind of compression, so part of the energy of growth is given to the mechanics. So you always have to understand that since you have in a nonlinear material, you cannot spend all the money that you get from the chemistry. Part you have to pay it to the mechanics. So at one point, it arrives at trade-off points where whatever you get more, you have to spend in mechanics. There is no longer money that you have left for growth. So this is actually an example. One other easy example, I will just skip very easy on that, is just the Drosophila wing disk, which is the paradigmatic example. In this case, we can imagine that there is no stress, there is no growth, but you can couple the chemical energy and the growth energy, and you actually fit very well with experiments that they have a fixed rate of conversion. So actually you can see that the morphogenetic fields and the growth, they scale over growth at the same rate with a fixed constant rate. Then there is another question on that I won't speak now, which is how does the tissue know where to stop? So and that's, I think, that mechanics has a role in that, because we have to convey global information, which is the size, with the local information, which is everything that the cells can have from the environment. So that's mechanics. I think it will have a bridge in that. We can even push this theory to a further extent by saying that we all know that there are chemicals that diffuse and stuff, but if you go and look at numbers, sometimes these diffusion fronts are very, very narrow, which means that actually instead of considering these narrow volumes as volume change, we can consider them as non-material interfaces. So actually an interface, a surface who carry thermomechanical properties. By doing so, we always have kind of homogenization of that, so we have the same balance laws, but for the surface fields, which actually take into account for the jump of the volumetric fields from one side or the other side. I'll just make a pattern example on that. But the important thing in here is actually, for those of you who are familiar for phase transformation, we have the fact that we have a Poynting vector, so we have an energy flux momentum which comes from chemistry and which is dependent on mechanics. Typical examples that we can construct for them are those self-similar forms that we've seen from the beginning, which are the seashells, for example, or, for example, the formation of horns, and etc. So it's just simple kinetic information on how we can construct these shapes. So now let's go into the application of that. How that we have constructed a theory which condensed in 15 minutes can give you even more envy of a coffee now, but we'll just try to see some nice pictures to see how this can be applied in few practical examples. So actually, it's a new branch which more than morphoelasticity, I will call it morphomechanics, because it just has a wider meaning of putting together softer liquids or solid materials, which has everything that puts together what is classically mechanics, so constitutive equations, whatever balance laws, and whatever we want to think about it, and those physical sciences, which are perturbation theories, singular perturbations, stability analysis. So how actually we can investigate the mechanical models to have results in terms of shapes, so in terms of some quantifiable stuff that we actually observe. And we will talk of emergence of shapes, because we don't think that shape is already there, but it's something that comes out as a dynamic feedback. One first example is growth of a colony of bacteria on a petri dish. So imagine you have a petri dish with some, you sputter some kind of sugars in that, and you just inoculate a round colony of bacteria. Okay? It's a thin geometry. You have a thin geometry, so it behaves more or less like a fluid environment. So actually, by classical scaling law, you have a Darcy law for the material. What happens? It happens that these nutrients are there, can diffuse from the outer border, and are uptaken only if by the bacteria. So outside here in the green area, they just have no uptake there, they can just diffuse. If they see a bacteria, bacteria see the nutrients, eat it. So in general, we have a volumetric growth term, which is the gamma, or a non-convective mass flux. And this we can just put a dependence on these terms on the local concentration of nutrients. What we observe in experiments, actually we observe a very different outcome depending on the kind of bacteria, conditions, etc. You can have some round colonies that keep on being round. You have some blebbing in some experiments, or you have some more complex that in some cases have like a fractal structure. So if we want to use continuous methods, we cannot describe fractal stuff, but let's see what's in the middle. Why certain have one morphology and others don't. So we consider actually two cases. In one, there is always only non-convective Mach flux. What kind of expression we get? Keller-Segan, so it's just chemotactic. On the other side, it's just something that we grow depending on, the more you have, the more locally you have nutrients, the more you grow. So volumetric source depending on n. So actually you have, while in classical growth problems, in Darcy you have a Laplacian of P being to 0. Now here you have a Laplacian of B, even the Laplacian of the nutrient, or just the local concentration of the nutrient. This changes a little bit the problem. We look for the quasi-static solution, we perturb it, and we actually construct the marginally stable Qs. And what we find that this solution is always unstable. So for those of you who are familiar with fluid instabilities, this reminds a lot of Suffman-Taylor, for example. Fingerings, that it's kind of clicked from the beginning. You kind of suspected some kind of similar mechanism. So we implemented infinite elements. We do only the fully nonlinear evolution of the system. Actually, we see that by only having those two non-dimensional parameters, which are what? How fast I grow compared to the chemical energy I get, and surface tension over friction. So mechanical parameter, growth parameter. I have only those two, and actually what we find is that the faster it grows, it stabilizes, which is something counterintuitive, but it's something found in fluid instability. And surface tension stabilizes, that's kind of stabilizes short wavelength, of course. So actually we see that these patterns, these fingering patterns, actually belong more to the chemotactic growth, while these blebbing patterns are more related to the volumetric growth. So actually it gives us an idea. So this was an example of fluid-like instability. How do I deal with solid-like instability? That's, I think, a nice example that we have done in collaboration with Ellen Kuhl and Stanford. We just imagine you have an endoscope and you put it inside your mouth. Don't do it yourself, make a professional do that. But what he will do is just that he will see that these wrinkles that you have in your gastrointestinal tissue are changes. The fact that we have circumferential wrinkles, zigzag, bilayers, ridges, longitudinal ridges, or whatever. But actually, very few of us know, and I didn't when I started that, and all of them originated at the same point in life, which is 2 weeks after fertilization of the embryo. So you already have your intestinal mucosa almost in an adult form after 2 weeks that you have been inseminated, which is kind of surprising. And all of them originates by 2 layers of tissues that starts growing at a different rate. So what happens actually? You can imagine that there are these 2 layers of tissues, that the orange one grows faster, which is the mucosa, grows faster than the outer one. So what happens? That actually you have a compression hoop stress in here, a tensile hoop stress in there, whenever they restore the compatibility. And by doing so, you can imagine, is there a point where this differential growth actually can destabilize this pattern, meaning that if we look for a wrinkled solution, this solution has less energy than the undeformed solution. How do we do that? We just, like we've done in fluids, but with a little bit more of mathematical complications, which means that we perturb the configuration, the spatial configuration, we construct the incremental problem, and we solve the elastic incremental problem. So we solve that, we impose incompressibility, all this, we are a living matter, most of matter is made of mainly of water, so it has to be incompressible. We solve it, there are different strategies, but actually we can construct some marginal stability curves. Now we'll see why these are important. So we actually imagine that there could be 2 patterns having wave number m and kz, which is hoop and longitudinal. So we want to investigate the whole stuff. So what we get actually, it depends on the ratio, on a geometric parameter, which is the ratio of the thicknesses, and on a mechanical parameters, which is the ratio of the shear moduli. This is a hint saying that we can demonstrate that if there is a marginal stability curve, means that the axisymmetric solution is no longer stable, but we don't know where this evolves. But we can use that as hints in numerical simulation to follow, to implement a continuation method to follow the solution in the fully nonlinear regime. And actually what we find is that all the morphological variety of patterns that we actually observe are completely controlled by mechanics and geometry. So the fact that at one point there is a bifurcation, we develop villi, intestinal villi, or plica circulares, or the folds that we have in your esophagus, depends actually on the fact that these have different geometrical and mechanical ratios. So the selection of the pattern is mechanical and geometrical. The evolution is just the fact that depends on the growth. So the chemistry keeps on growing, and the growth from the wrinkles evolves, creates creases or folds. At one point we even create, there is a enormous richness of patterns after that. There is wavelet doubling, tripling, quadrupling, jumps, subcritical bifurcation, supercritical bifurcation. It's a universe inside of that. But what it's known down, that's mechanics stops there. That's no longer the task of mechanics here. Because what it has been demonstrated is that once you create a pre-pattern, this pre-pattern actually has a feedback on biology. So actually, if you have those wrinkles and those folds, the cells which are on top of them feel the curvature and they differentiate. So they create other structures. They create the villi, glands, whatever. So it's an example of how the mechanical pre-patterns actually induces biological differentiation. These are just few applications that I won't speak here because we have heard a lot about tumors, but it's actually a strategy that we have in tumors as well. This is an work that we have done at NS in Paris with on skin tumors, and the fact that instability actually can have an escape mechanism for a vascular tumor to invade. And this is actually a brain tumor and actually a mechanical problem of the fact that protrusions in spheroids can be driven by mechanical instability. So another point, which has a new point, it's interesting, is the fact that from a mathematical viewpoint, you know that if you use free energy, which is polyconvex, strong elliptic, whatever you want, in tension, everything is regular. You will never have an instability. But you take a human nerve, you stretch it, and what you get, that after at a certain stretching point, you create these beds on a string pattern. Why so? the fact that the loss of stability here is by the fact that you add a surface energy, because you have a film on top of it. So actually you are able to create an instability by the so-called complementing condition. So you no longer, you have a loss of convexity at the border, at the free border. So actually it's some easy example, what is called now all elastocapillary. So let's put together elasticity and capillary energies. They scale different depending on the miniaturization. So there is a size at one point where they have the same order of magnitude. So they compete. By competing, the cylinder wants to be a sphere because there's a phase tension. The elasticity wants to create a cylinder, but if they have a typical comparable size, they can find a compromise and say, okay, let's make a pearl structure. The same point, we can see that this actually is governed by one ratio, which is the ratio of the elastocapillary length, so the ratio between bulk and surface energy, and the initial ratio. And depending on the applied stretch, you can actually change the morphology. By doing so, you can even push this analysis to a further extent, which means you make multiple scale analysis and you make weakly nonlinear stability analysis. Why do you do so? Because you want to, for example, understand if you have a continuous transition to a pattern or you have a jump. So in order to do so, you have to construct the weakly nonlinear stability analysis. You have your Ginzburg-Alangoa equation and you study it. And what actually you find is that in some cases, this is a continuous transition. In some other cases, this is not. There is a jump. So this is subcritical bifurcation, there is hysteresis, and whatever you find. But what you actually find in this is that if you put the amplitude versus the stretch or versus the elastocapillary ratio, you have the fact that at one point you can create a bedded pattern as you wish, because at one point here you have the amplitude is the same, but you can variate almost linearly the width of the pattern. In this case, you can create bubbles and just bed on stick. And this has a lot of applications. If you can think of stretchable electronics, you actually tune shape and function at the same time. So actually, I think it's a frontier now, in this case, to understand how pattern is related to the mechanics of the part. A last example, before going to take this well-deserved coffee, it's a modern idea or a provocation to most of you. But as I know that the mathematical community of angiogenesis, they only think of reaction diffusion equations, inhibitions, lateral inhibition, side inhibition, salt and pepper, whatever. What's angiogenesis? Angiogenesis for dummies like me. It's just you have a tumor at a certain point grows avascularly, but he is too big, nutrients don't penetrate, so it sends out some chemical factors. It's so-called the vascular endothelial factor. These factors reach a vessel, and there they promote the sprouting of a new vessel. So actually this vessel travels, reach the tumors, joins, sends the blood inside, so it changes its fuel. It's like babies when they move from having only the teeth and they start eating really solid food. It just changes the growth rate at one point. So if you see the patterns, the main idea was like, okay, it's true that there are an incredible, huge complexity of chemistry inside there. There is all kind of a reaction, inhibition, whatever. But what if this kind of structure somehow could be related to some known process like supercooling? What's that? Mullins-Kerkel stability, you supercool. There's what is called dendritic growth. Why not? Why not just proof of concept? Let's implement a model like that and let's see if it works or not. So we have the tumor here. The tumor sends a signal. Here it's a sphere. It will send like a spherical wave. Spherical wave at one point will find the capillary. The capillary is so small that we can consider it as a non-material interface. So it travels, reaction diffusion. It sets somehow, he has a jump in the gradient because he has an uptake here at the non-material interface. And this non-material interface, we can construct a law, a mechanical law, which make it evolve depending on the chemistry. And we had even one more equation which reminds of the Gibbs-Thompson equation, is the fact that the chemical potentials there depends on the curvature, because it's something actually observed in cells. So we have only 4 non-dimensional parameters in that, and he doesn't want to go further. So we plug it into a 2D model, so that what you see here is the tumor boundary. At one point there is this vascular endothelial factor, and this is the capillary wall. The capillary wall is here. At a certain point it gets a threshold concentration of the vascular endothelial factor, and it starts sprouting. Sprouting, it follows those laws, which are mechanical, and what we get is just that actually we have kind of branching patterns that actually evolves towards the tumors and eventually reach it. So this is, I don't claim this is an answer, that this is like a mechanics of concentration, but I think it's just a proof of concept. It might work. Now, will we be able to put chemistry, more complicated chemistry, in a simple mechanical model? Well, we'll see. I think we am to an end. I just want to thank you for your attention, and just thank all my collaborators, especially David and Luigi, and Chiara and Matteo, who have been wonderful postdoc with me, and Ellen for sure for this, for the work for the gastrointestinal system. I will be able to answer, will be very happy to answer to all your questions here or during the coffee if you prefer, if time allows. Thank you.
Well, we've just started a few minutes late, sorry, so we've got time for a couple of quick questions if there are any.
So for your gut modeling, did you have to use anything anisotropic, or you were able to go the whole way with an isotropic?
Which model, sorry?
Gut intestine.
The intestine, actually, we started with an isotropic model. What is
Isotropic?
Isotropic? No, it's only isotropic. We don't. When I can, I always try to put less ingredients as possible. I don't want to add complexity. If I can have a good shape with the simplest model, I'm happy. What is known after is that, for example, zigzag, they depend on anisotropic growth. So at one point it's just growth that changes polarity. At that point they are able to do secondary bifurcation. But for this kind of selection, only isotropic is okay.
The images of the patterns in the intestines that you showed, you had these, this progression. Was that a progression in time, or does each of those represent a steady state on a domain that's growing?
No, it's actually, there are a number of studies in embryos where they actually, you can see it, all of them happens normally at days 13, and they go towards a steady state in a couple of days. But every steady state for each part of the gastrointestinal system is different. So actually it's really, in my case, it's just growth that happens slowly. So at any time that growth happens, the viscoelastic response is very quick. So it's a sequence of steady states from an elastic viewpoint. But it's in biological realm, it happens in a couple of days.
Okay, well, I think it's time for some tea. So would you join me in thanking both our speakers again, and we'll be back here at 12:30.